Good evening.
From all around the world. I always say to Andrew, I never see a turnout for events as I do for Humanists UK. So thrilled to welcome you. And we all know this is a particularly monumental lecture. Marking International Women's Day, this lecture series explores and celebrates the contributions of women towards the promotion and advancement of aspects of humanism in the UK and around the world. The Rosalind Franklin Medalist has made a significant contribution in one of these fields. The lecture and the medal are named, of course, for Rosalind Franklin, humanist and scientist, whose contribution to science for many years went unacknowledged on account of her sex, and who is today rightly celebrated. We do want you to ask questions. There'll be lots of time to put them to our speaker a little bit later. Don't submit them yet because it can be distracting, but there is a Q&A tab. I'm sure many of you are used to it now on Zoom calls. And so save up your questions, and when we finish the lecture and I start to ask a couple of questions of my own, that's the moment to start submitting them, and we'll take them as many as we can. Put your name if you want me to say who you are and give you a name call when I use it. Just before I introduce Sarah, I'll take a moment to remind you again that the hashtag for tonight is hashtag Franklin Lecture. So do feel free to tweet about it and share it on social media. So our lecturer tonight is Professor Sarah Gilbert, Professor of Vaccinology in the Nuffield Department of Medicine at the University of Oxford. She completed her undergraduate studies at the University of East Anglia and her doctoral degree at the University of Hull. Following 4 years as a research scientist at the biopharmaceutical company Delta Biotechnology, she joined Oxford University in 1994 and became part of the Jenner Institute there when it was founded in 2005. Her chief research interest is the development of viral vectored vaccines that work by inducing strong and protective T and B cell responses. And she works on vaccines from many different emerging pathogens, including influenza, MERS, Lassa, Crimean Congo hemorrhagic fever, and SARS, working with colleagues in the Jenner Institute Research Labs, the Clinical Biomanufacturing Facility, and the Centre for Clinical Vaccinology and Tropical Medicine, all situated on the Old Road campus in Oxford. She's able to take novel vaccines from design through to clinical development, with a particular interest in the rapid transfer of vaccines into manufacturing and first in human trials. And Lord knows, rapid transfer and the rapid development of vaccine is one of the amazing stories of this past year. She is the Oxford project leader for the key vaccine that's been developed against the novel coronavirus. It's been used to vaccinate millions of British adults in the first weeks of the national vaccine rollout. I'm thrilled to welcome the Rosalind Franklin Lecturer tonight, Professor Sarah Gilbert.
Thank you very much for the introduction, Samira. So I'm going to get straight into my talk, and I'm going to give you a bit of background leading up to what I did before 2020 to try to set the scene a bit so you can maybe see how we were able to go so fast when we got to 2020 and really needed to make a rapid response. So first, I want to start off by reminding you that emerging infectious diseases have to come from somewhere. They don't just spring up fully formed from nowhere. Most of the time, they're actually present in different livestock species. And most of the time, they stay in those livestock species or wildlife, and we don't really notice what's going on. There's actually an awful lot of viruses out there that we probably don't even know about yet, and they don't need to worry us because they stay with the species that they normally infect. But occasionally, we get what we call a spillover. So occasionally, a virus will move from, say, a bat into a camel, and then possibly people who are in contact with a camel gets infected. We might have influenza viruses moving into pigs, moving into domestic poultry, and then that means that we're more likely to be exposed to the virus. So often the turkey or the duck isn't the original source of the new virus that starts to cause infections in human, but it gets amplified in that intermediate host. And then because people keep the turkeys or the ducks and they slaughter them and they get exposed to the fluids from the animal, then they can get infected. So occasionally what we do see is viruses starting to infect humans when they hadn't been infected humans before. Now, one of these is Ebola, and you may remember the 2014 Ebola outbreak in West Africa. It may be something you've forgotten about now. It was something that I followed in the news every day, as we did with the COVID outbreak last year. We were following what was going on in West Africa with Ebola in 2014. This actually is another viral disease. It's a hemorrhagic fever. It causes a lot of fever and a lot of bleeding, blood loss, high fatality rate. And in 2014, this virus, which is normally found in bats, started to infect people. In fact, it probably first happened in 2013, this particular outbreak. Now, this particular virus has been known about since 1976. 2014 wasn't certainly the first time it had ever caused outbreaks in people. But most of the outbreaks before had been in quite isolated rural areas where people were in closer proximity to bats. And they tended to have been dealt with fairly quickly by the tried and tested methods of contact tracing and quarantine. So finding out if somebody is ill, who they've been in contact with, and making sure that those people quarantine. And when this was involving fairly small numbers of people in rural areas and they could be well separated, that was an effective method of controlling the disease. But what happened in 2014 was that the disease slowly spread, and it rumbled on, and then it moved into cities, and then we had high population density, lots of people being infected quickly, more difficult to do the contact tracing, and it grew into an outbreak which spread across 3 countries, in Guinea, in Sierra Leone, and in Liberia. And we started to see cases popping up in small numbers in other countries when people traveled. Now, these borders between the different countries are quite porous. People go back and forward, and you really have to treat the whole outbreak area as one. And there was no vaccine. There had been work in vaccine development done mainly in the US as part of a biodefense program, because this is such a scary virus that there had been concern about it being used as a weapon. And so vaccine development had happened so that if that was to take place, we might have some possibility of defending people against it. But this vaccine, which was still at quite an early stage of development, wasn't being used in West Africa, and gradually the case numbers grew. These different colours represent the different countries. Most of the cases in Liberia, case numbers increasing rapidly through 2014. This was really getting out of control, and the old methods of containing the outbreak just weren't working. So there were 2 vaccines that had gone some way in vaccine development. And with myself and other colleagues at Oxford, we got involved in a consortium to try to really accelerate the vaccine that had been developed by a company called Kairos. They were using a viral vectored vaccine. It was an adenovirus that normally circulates among chimpanzees rather than humans. That means humans don't already have antibodies to it, which is useful. And they'd use this to make a vaccine against Ebola. And we'd worked with the company on some malaria vaccine development and vaccine development against other diseases using the chimpanzee adenovirus, or CHAD3 vaccine platform. But in 2014, we got together with a group of other people to try to really accelerate vaccine development for this. It had been tested in animals previously. A batch had been manufactured, ready to go into clinical trials in humans. It hadn't been put into the vials, ready to initiate that trial, and that's where things had stopped. So we were asked to help get this vaccine put into the vials, tested, do the applications to start a clinical trial, and we knew we could do that very rapidly in Oxford. And our plan was to do a phase 1 trial of this new vaccine against Ebola in Oxford, starting in the middle of September with 60 volunteers. That would provide data on the safety and the immune responses coming from the vaccine, which would then go into a phase 1 study in Mali. Now, this is another West African country. It was right next to the outbreak area, but they didn't have the outbreak in Mali. However, the healthcare workers were happy to volunteer to take part in the trial, because if it did spread into Mali, they would be the ones who would be needing to respond to the outbreak. And in parallel with doing these trials, manufacturing of a larger batch, 20,000 doses, was set up. So the objective was to get safety data from testing in 140 people in the UK and in Mali, particularly in healthcare workers, and seeing if the immune responses that were generated were similar to the levels that had been previously used to protect monkeys when the monkey studies had been done. And that was then expected to lead into a decision whether to carry on with the vaccine development and do a larger phase 3 trial to test if the vaccine was effective. Does it stop people getting Ebola infection? And we thought that that might be possible to start by December of 2014, again, mainly targeting the healthcare workers who, as with COVID, were becoming infected in the course of their work, and also could then spread the disease to their family and to other patients. And so they were the most important population to target first with vaccination. So this was a very rapid timeline for that clinical trial. The grant application was submitted to the Wellcome Trust, who actually helped write it rather unusually, on the 14th of August. And that funding was awarded only 12 days later. It can, in normal times, take anything up to a year or even 18 months to get funding for a project of this nature, but this was done very rapidly because of the need to move quickly. The vaccine had been made, it was a bulk product in a large quantity, but it needed to be put into the individual vaccine vials before it could be used in the clinical trial. That was done. The application to the MHRA was submitted so that the trial could legally take place, and there was an ethical application as well. We need both regulatory approval from the MHRA and approval from an ethical committee to be able to start a new clinical trial. Both of these were prioritized and went very quickly. The vaccine was delivered, the labels were put on. We had these very complicated labels for vaccines being used in a clinical trial. It tells you a lot of information about what's in there. And finally, a contract was signed for the funding for the trial. And on the 17th of September, the first person in Oxford was vaccinated. And by the 18th of November, 60 people in Oxford had been vaccinated, and the data was being provided to Mali so that the clinical trial could start there. Now, while this was going on, the outbreak was still continuing, and we were starting to see over time the numbers of cases were declining as we got through to the end of 2014 and into 2015. Although we did our phase 1 trial, we got very good safety data, we showed the immune response of the vaccine, and then the trial in Mali started. There was then a delay over the plans for the phase 3 trial to take place in West Africa. There was discussion about whether a placebo-controlled trial, which is the gold standard for testing vaccines, was ethical because this is a disease with such a high fatality rate. What should be the best design of the trial? While those discussions were happening, the numbers of cases were declining. And in the end, of 2 vaccines that went into early clinical development, only one of them was tested in an efficacy trial. And it wasn't the one that we'd worked on, and unfortunately for us. It was a vaccine which was shown to have very high efficacy, so it was a great success. The difficulty with it is that it requires really low temperature storage, so minus 70 degrees storage, and that's very difficult to use a vaccine that needs such low temperature storage in West Africa, where the ambient temperature is very high. But it wasn't possible to go on and test the vaccine that we'd worked on, because by this time the outbreak had been contained, and this wasn't due to use of the vaccine, it was due to just persisting with the contact tracing and quarantine method. However, as a result of this, the world did have a first Ebola vaccine that was licensed then for emergency use. Another one has been developed subsequently. But although our early vaccine testing had gone very quickly, it then just ground to a halt when we had to talk about how to get into vaccine efficacy testing. So that was a learning for those of us who are looking to do rapid vaccine development, that you need to have all parts of the plan to test the vaccine ready to move quickly. It's not enough to do a phase 1 trial and say, yes, this vaccine's safe and we get a good immune response. You need to work out how you're going to test if the vaccine is effective and be ready to do that really fast. And that kind of thinking was behind what we did in 2020. So let me tell you a bit about the technology we use, the way we make our vaccine. And this is a cartoon on the right-hand side here of an adenovirus. So there are lots of different adenoviruses. Most mammalian species will have adenoviruses that will infect them, and some can infect more than one. We use an adenovirus that's also normally found in chimpanzees rather than humans. It's not the same one as in the Ebola vaccine trial. It's one that we developed in Oxford. But the fact that it doesn't circulate in humans normally and cause respiratory tract infections, as adenoviruses do, means that we don't have any antibodies to it. Now, there are lots of closely related adenoviruses that do infect humans. They cause a cold. We don't generally know that we have an adenovirus infection. We just know that we have a cold, and it's a fairly mild illness. But we get infected with adenoviruses all the time. It's quite a common thing to happen to us in the winter. And those are adenoviruses that have all of their genes intact, and they can spread through the body and cause an infection. But we don't want that to happen with our vaccine. So what we've done is removed some of the genes from the adenovirus. And then what we do is add in a gene that codes for, that provides the instructions for the extra piece of protein that we want to make. And in the case of nCoV19, it's the spike protein from the coronavirus. So we add DNA that gives the instructions to make the coronavirus spike protein into our adenovirus vector. And having taken out some genes that mean that the adenovirus can't spread through the body after we use it to vaccinate somebody, it's replication deficient. And we've been using this approach for a while, using the ChAdOx1 that we developed since we've had more than 330 subjects vaccinated in clinical trials with different antigens added into it before we started the SARS-CoV-2 trials. These were different antigens, and I'll tell you about some of them. But more broadly, simian adenoviruses, the CHADs, have been used in over 6,000 people, in infants in Africa, in malaria vaccine trials, and in older people in the UK in flu vaccine trials. And we saw that we got a consistent safety profile and strong immune responses after one dose. And importantly, for the COVID vaccine development, I'd also started working on a trial against another coronavirus, MERS, Middle East Respiratory Syndrome. And I'll again tell you a bit more about that later. So this is a cartoon of the virus, and it looks quite a scary thing. It's not really in different colors. This is just to help you see the different parts of the virus. And I was reading something recently about a GP who'd been saying that he'd been talking to one of the members of his staff at his practice, and this person didn't want to take the COVID vaccine. And he had quite a long conversation with this member of his staff. And at the end of it, said that, well, she just felt that it was something foreign, and she didn't want something foreign in her body, and that was that. And my response to that is, well, of course it's foreign, otherwise it wouldn't work as a vaccine. So we have a really complex and highly evolved immune system. And what the immune system does is continually scan the body for something that shouldn't be there. So we call it distinguishing between self and non-self. So when the immune system's working properly, it doesn't attack our own bodies. That can happen in autoimmune disease, but when it's working properly, that doesn't happen. What it's doing is looking for something that's not us, looking for something that's foreign. And when it does, it will spring into action. It will start to make antibodies, it will start to make T cells and deal with that invader. That's the job of the immune system. Now, when we make a vaccine, we want to stimulate the immune system in that way, but we want to do it in a way that's safe. So I told you that this adenovirus vector that we use can't spread through the body. We use it because it carries the gene for the coronavirus spike protein into the body when we inject it into somebody's muscle. It goes inside their cells, as an adenovirus normally will do, and it starts to make large quantities of that spike protein, and then it stops. It can't go any further. If we don't make any immune response to it, it still can't go any further. It's disabled, it can't spread, it can't do us any harm. It will induce an immune response, and that immune response can give us some side effects. It can give us headache, fever, chills, sometimes pain at the injection site. These are all quite short-lived things that can happen after vaccination because the immune response is developing. And as a result of that, we get left with an immune memory. And that means that if we then encounter, in this case, the SARS-CoV-2 coronavirus, we've already got the immune memory that suddenly activates and springs into action and protects us against that coronavirus infection. Now, any viral infection is something foreign coming into the body. Any bacterial or parasitic infection is something foreign. And what our immune system does is find the things that are foreign and destroy them. And we train the immune system to do that by using a vaccine. And I think it's a bit like if we're training our muscles, if we're running or if we're weightlifting, we are training the muscles to be bigger, to be stronger. We can have some temporary soreness of the muscles, but as a result of the training, the muscles will grow, they'll become more effective, and then we're better able to do the task that we want to do. It's a bit like that with a vaccine. We're using it to train the immune system in a controlled and in a safe way without giving somebody a real infection. So for me, saying that you don't want to have a vaccine and you'd rather take your chances with the viral infection itself is a bit like signing up to run a marathon and then not doing the training for it. And you're really going to feel how difficult and stressful it is when you actually start to run that marathon if you haven't prepared, if you haven't done the training. So the vaccine is giving your immune system the training, it's laying down that immune memory so we can then respond. And in order to do that, it has to be something that's foreign to the body, but it's not there for very long. It's there for a few days, it does its job, and then the immune memory is there and the vaccine has left the body.